Hi, everybody. Welcome back. It's Dr. Hall, and we're going to continue lecture number 2 on the cells of the nervous system, focusing now on neuroglia. So if you remember our outline, there are 5 types of neuroglia that we're going to talk about in this order. So let's get started. A quick reminder that the other cells found in nervous tissue would be the neurons. That's what our previous part of the lecture was about. And neuroglia are everything else, so named because of the Latin word for glue, because they didn't know what they did initially. And now we know that there are 5 main types of them that we're going to talk about in a minute, and they are way more numerous than the neurons are. So they carry out several important functions, as we'll see. Here we go. Just a reminder, they are cells. So like all cells, they have all of the organelles that you would expect, including a plasma membrane, which controls what goes in and out. There are going to be receptors on those membranes. There are going to be ion channels on those membranes, things that are going to become important later on in the class. So just always keep that in mind. I also liked this image from the journal Nature that shows a few of the different types and some of what they do. The first The first cells we're going to talk about are astrocytes, which the name actually translates to star cells. So astro, like astronaut, that word actually comes from Latin for star. So it's kind of lovely to think about. So somebody thought they were star-shaped cells, hence astrocytes. Astrocytes are the most numerous of all the glial cells. They are found throughout the central nervous system. As I mentioned, they are star-shaped, and they have 3 main functions for our purposes in this course. One has to do with the blood-brain barrier, one with the tripartite synapse, and the other one has to do with trophic factors. So we're going to go through all of those one by one. I do encourage you to come back and look at this graphic later, though, after we do, because then it's going to make some more sense. So let's get started with those. Okay, so blood-brain barrier. You may remember, hopefully, that in the central nervous system, there is something called the blood-brain barrier. This is a special protective structure that makes it harder for things to move out of the bloodstream into the brain tissue than it is for things to move out of the bloodstream into tissues in other parts of the body. So those could be molecules, those could be drugs, and of course, those could be germs or pathogens, bacteria, viruses, things that can do you harm. So it's a protective structure that's composed of some interesting features. And one of the features of the blood-brain barrier is due to astrocytes. So astrocytes, as you can see in this diagram here, let me see, maybe purple will show up. Well, it uses purple, but anyway, you can see these little projections coming out from the astrocyte. I'm going to change my color here to green. All right, and you can see this one here is wrapped around a blood vessel. So now if we zoom in, you can see here in the middle, right, is our blood vessel, our capillary with red blood cells in it. And these little pink cells here are the endothelial cells, which are the cells that make up the lining of the wall of the capillary. And then you see these astrocytic end feet. So astrocytes send out these projections, and then they make this little foot shape, and it kind of forms an extra layer around the capillary to help protect the brain from things that might be in the circulatory system. Now, obviously, there are important things that need to move from the brain or from the circulatory system into the brain and vice versa. So, of course, we're going to have mechanisms for those. But remember that oxygen and carbon dioxide can freely pass through plasma membrane. So that's not going to be an issue there. So job number 1 of the astrocytes is to contribute to the blood-brain barrier with their little end feet that they put out and they put around the walls of the capillaries, which is just kind of really cute. Job number 2 is this tripartite synapse. So a synapse is a place where one neuron connects with another. Hopefully you remember that from a previous course because we haven't talked about it really yet in this class. And tripartite just means that there are 3 parts. So tri like tricycle, right? So some synapses have an astrocyte associated with them. So here we can see down here an astrocyte sending a little projection that's kind of holding those 2 parts of the synapse together. And what's interesting is that these astrocytes can have receptors for certain neurotransmitters. They can have ion channels for things like potassium and sometimes chloride or calcium. So these astrocytes can modulate the synapse by changing what happens to neurotransmitters, changing what happens to ions. And when we start talking about signaling between cells in the next couple of weeks, you'll see that this is going to be really important. So the astrocytes, job number 1, blood-brain barrier. Job number 2, modulating synapses as these 3-part or tripartite synapse. It's a 3-way. All right, last big job of the astrocytes is trophic factors. Now, what are trophic factors? They're just things, molecules that are produced by the astrocytes. They can do various helper functions. They can encourage the surrounding neurons to do different things. So it can signal them to keep living, keep going, don't give up. It can signal synapses to continue. So one of the ways that our nervous system changes, remember we talked about plasticity before, is in formation of synapses, either forming new ones or getting rid of old ones. So those trophic factors help in that process. And they can also guide where those synapses should form, those new connections should form. Astrocytes with their trophic factors also help with repair after injury and say, oh, over here, you need to grow over in this way to replace the part that was damaged or lost. So that's the third function of astrocytes that we're going to focus on in this lecture. So just remember, they do a lot. So these are 3 really, really big categories of things, and we kind of glossed over modulation of synapse. We kind of glossed over trophic factors. So this is a graphic that I found from a study from not too long ago, looking at how astrocytes are involved in repair and also sometimes some of the damage that occurs after somebody has a stroke. So under normal conditions, when everything is good, we would call those physiological conditions, right? That axon guidance and growth, right? Guiding where synapses should be. They ECM modulation, which means extracellular matrix, neurogenesis, which is that crazy idea of growing new neurons. They regulate the blood-brain barrier. They help with a lot of different things through those trophic factors. And then you can see under pathophysiological conditions, so conditions, those are disease conditions. Pathology is when something goes abnormally wrong. So all types of things. Now, do I want you to memorize all the things on this diagram? No, not at all. I just want you to understand that astrocytes do a lot. They're really important, and that's the reason why they are the most numerous of all the glial cells. They're kind of a big deal. Now, unfortunately, astrocytes, just like any cell type in our body, can become cancerous. So any cell type in your body can become cancerous. What it means when a cell becomes cancerous is that it stops behaving normally and starts doing things that it shouldn't. They tend to not die, they tend to replicate really quickly, and they tend to move into places where they don't belong. So all of those things are true of any cancer. Cancers are generally named for the type of cell where they started. Did they start in a cell in your colon? Did they start in a cell in your lung? Or if it was an astrocyte who all of a sudden went bad, then that would be an astrocytoma. Now, interestingly, then because they're the most common glial cells, this is the most common type of glial cell tumor that we see in people as an astrocytoma. Some of them, especially the ones that occur in children, tend to be relatively benign, but then there are some that are really aggressive and have a very high mortality rate, and those are called glioblastomas. So if you ever hear of glioblastoma, that is actually a very aggressive form of cancer of an astrocyte cell. Next, we're going to talk about oligodendrocytes and Schwann cells. Now, those are 2 different types of glial cells, but their functions are almost identical, so we're going to talk about them together, kind of like Tweedledee and Tweedledum, if you know who that is. So these cells produce the myelin sheath. So let's remember the myelin sheath is that fatty insulating layer that covers the axons of most neurons, not all, but most. Remember, it is not a continuous sheath. It is not a continuous covering. It's in little sections or segments, and those places in between are called the nodes of Ranvier. So let's all review from the first half of the lecture. Now, the key thing to remember is that the oligodendrocytes are found in the central nervous system. So if we're in the brain or the spinal cord, it is the oligodendrocytes that are producing the myelin, whereas if we're out in the peripheral nervous system here, then they are Schwann cells. So they do the same function, but they have different shapes. Let's take a look. So oligodendrocytes are really weird. So you have this central part of the cell right here, and it's got its nucleus, and then it sends all these projections out in all these different directions. Okay, red probably would have looked better here. So, right, so it's sending all these projections out, and then they wind around sections of axon to produce segments of the myelin sheath. So it's a pretty good diagram. You can get an image here. Now, this diagram only shows like 3 projections, but oligodendrocytes can have 50, or they can have tons, right? So in the central nervous system where everything is packed and there's lots of axons really close together, there's not a lot of room, you have one cell that's like, putting these little myelin wraps around segments of lots of different nearby axons. So what's so interesting about oligodendrocytes is that, those extensions of the cell that are wrapped around axons can be 100 times the size of like the cell body itself. And maintaining that is a lot of work. So apparently they're like one of the most metabolically active cells in our brain. So they're ones using the most energy just to maintain the myelin. It's important. All right, I think of these guys kind of like Elastigirl. Like I think of like her arm going out and wrapping around an axon, and her other arm going out and wrapping around an axon. So that's kind of how I conceive of them. But I'm weird. That may or may not work for you. All right, out in the peripheral nervous system, we're going to have Schwann cells. So Schwann cells are entirely different in terms of their shape. So one single cell wraps itself around a section of an axon, and that's it, the whole cell. So as you can see in this diagram here, let me change to red. So you can see in this diagram here, that's the nucleus of the cell. This is the whole entire cell. It wraps itself around like a crazy scarf, and there you have it. So that's how you form the myelin sheath in the peripheral nervous system. Now, what's interesting about Schwann cells is if an axon is damaged, then a lot of times the Schwann cells are really important in kind of like establishing a little highway and being like, here, axon, this is where you grow, kind of like creating a little tunnel and guiding it. So that's kind of neat. You can see an image in the textbook on that as well. So that's oligodendrocytes and Schwann cells. One makes the myelin sheath in the CNS, the other makes the myelin sheath in the peripheral nervous system. Oh, so yeah. Anyway, microglia are next. So microglia are not really nervous cells at all. They're actually technically immune cells, but they live only in the brain. So you could think of them as nervous cells because they're not found anywhere else. And they are like those little robot vacuum cleaners kind of that go around your house. So they clean up things for us. So they will scavenge, meaning kind of like suck up and eat and get rid of dead and dying cells, cellular debris, clumps of misshapen protein, which is going to be important for some disease processes, other just garbage that's around in the brain, as well as pathogens, which is the fancy name for germs. So things that could cause infection and cause harm to us if it gets past the blood-brain barrier, because maybe the astrocytes weren't doing their job, then the microglia are going to come in and suck up and chew up and destroy those harmful substances. Last is ependymal cells. So these cells line the inside of the fluid-filled spaces in the brain that are called ventricles. Maybe you don't remember what those are. We're going to do another slide in a second. They also produce and circulate the cerebrospinal fluid, the fluid that fills those ventricles. So let's take a look. So inside of our brain are actually these fluid-filled spaces called ventricles. So you can see in this diagram here from the Hedges textbook, it's pretty extensive, this network of little kind of pools of fluid that are found inside our brain. And they actually, even this fluid, you see this down here, this central canal, it's actually going to continue and go all the way down through the center of the spinal cord. So we have fluid in the center of our brain, we have fluid in the center of our spinal cord, and that is the CSF, or the cerebrospinal fluid. Not only is that fluid there to help cushion and also kind of float the brain, because the brain's really heavy, and so we put some fluid in there to help kind of float it, give some cushioning, but it also circulates. So this fluid doesn't just sit there, it actually is continuously circulating through the system, which then allows it to kind of clean things out and move things around from one place to another. And so those ependymal cells, which are lining the insides of the ventricle and producing the CSF, also have my favorite organelle, which is the cilia. So cilia are these little hair-like projections that stick up from the top of the cell, and they beat. They go whoosh, 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 right? And so that beating of the cilia creates the movement of the CSF through this whole ventricular system. And when we talk more about the anatomy of the brain, we'll talk about that whole circulation of the CSF. So ependymal cells are pretty cool because they make the fluid and then they also circulate it with their cilia. So that's it for the 5 different types of neuroglia. So our astrocytes do a lot for us. Those end feet contribute to the blood-brain barrier, helping to protect the brain from substances in the circulatory system, in the bloodstream. They also can modulate synapses at those tripartite synapses. They can kind of say, excuse me, may I join you? And they help modulate what's happening. And then they produce these trophic factors, which are really important for cell health and growth and healing. So super important. Oligodendrocytes and Schwann cells, they make the myelin sheath. It's oligos in the CNS, Schwann cells in the PNS. And then microglia, which are immune cells that scavenge up debris and germs to keep everything nice and clean and healthy. And then the ependymal cells that line the ventricles, produce and circulate the cerebrospinal fluid. All right, that's it for lecture 2. Thanks, everybody.